So as we get ready to start talking about the phases of mitosis, it's important to understand that the cell that we are beginning the mitosis process with is a cell that is just coming out of G2 of interphase. So here we have a look at a cell that just came out of G2 and a few things to notice about it. The chromosomes are not visible. They are certainly there, though, and these are going to be duplicated chromosomes. And they are duplicated because they were copied during the S phase. So that means that these do look like little X's if we were able to see. They are made up of 2 chromatids each. And then another thing that I do want to point out here is that we are going to have 2 centrosomes. And the centrosomes in this picture I've shown in yellow. And these are going to be involved in producing the spindles that will be actually pulling apart the chromosomes a little later on. So the centrosomes happen to be part of the cytoskeleton of the cell. So if everything checks out okay and the cell decides to proceed with mitosis, the first phase that we're going to go through is going to be prophase. So let's talk about the things that happen in prophase. In prophase, one of the things is going to be that the centrosomes begin to move apart. And so as they move apart from each other, they're going to be moving towards opposite sides of the cell. So if I draw my cell here, and I'll put my nucleus back in, along with the chromosomes that are there, we're going to see that the centrosomes get further apart from each other. So they're spreading out, as we see right here. And as they spread out, spindles, which are again part of the cytoskeleton, form between the centrosomes. So we actually see them start to grow out of the centrosomes. So if we draw some spindles here, I'm going to show them in blue. So we'll actually see them kind of reaching out towards each other. And this is going to occur as those 2 yellow centrosomes continue to move apart. Other things that happen. One very important thing that's going to happen is the chromosomes start to condense. And so they are going to start to really wind up and get shorter and shorter and shorter. We do have special proteins that are going to be involved in that. And those proteins are what we call condensins. And so once the condensins get going, what we're going to see is that inside of the nucleus, we'll notice that we start to get these dark lines or darkly staining chromosomes. And again, that's indicating that we're getting a lot of winding up with our chromosomes so that they become shorter and thicker, indicating that we're just looping them up more and more with the help of those condensin proteins. One other thing that's going to start to occur here is that the nuclear envelope, so the membrane around the nucleus, begins to disintegrate. So if we show that here, what we're going to see is that we start to get holes in our nuclear envelope. And as soon as we start to get holes in the nuclear envelope, we will ultimately see the chromosomes start to spread out more into the whole center of the cell because there is no envelope or no membrane that's actually holding them together in that tightly kind of circular spherical structure. So that would be prophase for us. We're beginning to form the spindles. We are moving the centrosomes apart from each other. The chromosomes are starting to become visible to us, so we can start to see them because they are condensing with the help of the condensin proteins, and the nuclear envelope is going to start to fall apart. Once we've done all that, the next step is going to be prometaphase. So if we take this prophase cell and we start to move it into prometaphase, what we're going to find about prometaphase is that that is going to continue a lot of the processes that began in prophase. So if we begin to list some of these processes, we are going to, first off, we're going to continue to condense the chromosomes. As we do that, they are going to become more and more obvious to the eye if you're looking through a microscope. the spindles continue to form. And as they continue to form, the centrosomes continue to move apart. And they are going to continue until they reach opposite sides of the cell, and we would call those opposite poles of the cell. And then another thing that's going to happen is the nuclear envelope completely disintegrates. So the nuclear envelope really disappears. Okay, so if we begin to draw this, we've got a cell here. We now have certainly shorter, thicker chromosomes that are scattered around the cell. So our chromosomes in the cell, they are now scattered around, but these are going to be X-shaped chromosomes. So that's what I'm drawing right here. We can tell that they're X's now because they are so highly condensed because of the condensin proteins. And again, they're scattered around because there's no nuclear envelope there anymore. At this point, the centrosomes will be on entirely opposite sides of the cell, and they will have spindles coming from them. But one of the things that's going to also occur in this phase that has not been mentioned yet is that the spindles are going to attach, and they're going to attach to each sister chromatid. So we will see that a spindle actually attaches to here, and we'll have a spindle from each side, if I just reach some of these over, that attaches to each chromosome. Okay, so once we've attached these spindles to the chromatids, we're going to be able to actually move the chromosomes around quite a bit inside the cell. Now we do have a special protein structure that's located at the centromere where the spindles are actually going to be attaching. That protein structure is what we call a kinetochore. So kinetochore is the place where the spindles attach. And there will be one of these per chromatid. Okay, so each place that I've drawn this little connection in here, you would have a little kinetochore that the spindles are attaching to. So I'll draw those in like that. So that's prometaphase. So what we've done is we have continued to condense the chromosomes. Now we can see them really well. We can tell that they are X's. The centrosomes continue to move apart to the two opposite sides of the cell, and they have the spindles formed between them. And then the spindles are going to actually attach to the individual sister chromatids at the kinetochore, which is located at the centromere of each one. The very next phase, after prometaphase, is going to be metaphase. And metaphase is all about now moving the chromosomes around inside the cell so that we will be able to carefully distribute them later into these two new cells or two new nuclei that we're going to make. So in metaphase, if I draw my cell right here, we have the centrosomes at the opposite sides, and they've already attached to the chromosomes themselves. And so what's going to happen is that we will start to see the chromosomes get kind of lined up down the middle of the cell. And they will get lined up because we're going to have special adjustments made with the spindles that are attached to each one. Okay, so you have lots of spindles coming out of the centrosomes. Some were already attached to those chromosomes, and because of that, we can line up the chromosomes. So a lot of times when we do this, we say that this little place where they are lined up, we call that the equator of the cell. Or sometimes it's called the metaphase plate. Okay, but either way, we've got this distinct line down the middle of the cell where these chromosomes, they are oriented so that if we were to pull them apart, one sister chromatid is going to move towards one centrosome and the other chromatid is going to move towards the opposite centrosome. In the very next phase, the next phase is going to be what we call anaphase. In anaphase, we're going to see that our cell begins to become a little bit more oblong in its shape. So it's not the nice spherical shape that perhaps it was before. And what we'll see is that the chromosomes are going to begin to move towards opposite sides of the cell. So I'm going to draw it like this, and then we'll talk about what actually happens to move these apart from each other. In anaphase, the cohesin proteins are going to start to disintegrate, or in other words, they degrade. And about the same time, the chromatids separate. And when the chromatids separate, we can see that now we've got this movement of the chromatids in two different directions. And that is the result of the spindles kind of tugging on them and pulling them towards those centrosomes, which are on the opposite sides of the cell. As long as they're still moving towards the poles of the cell, we say that the cell is in anaphase. As soon as they reach the two poles of the cell, that would be when we end up in telophase. So our very last phase is telophase. And so if we draw telophase over here, telophase is going to take, and I'm going to draw it for the moment all in one cell here, but in telophase we have these chromosomes now at opposite ends. And now the chromosomes are, they're chromosomes made up of one chromatid each. And again, they're in different places on this, so opposite sides. And now really we finished the actual pulling apart of the chromosomes and we want to rebuild everything. So we still have our centrosomes here, but they've kind of done their job. And so things that we'll start to notice is that first off, the spindles degrade. So since they degrade or disintegrate, we don't see them anymore. They go away. Another thing that's going to happen is we start to reform the nuclear envelope. So we'll start to see that appear around our two groups of chromosomes. So we rebuild the nuclear envelope. And another thing is going to be that the chromosomes start to unwind. And what that means for us visually is that the chromosomes begin to kind of disappear, or we're not going to be able to actually see them. That's not to say that they're not there. They're there. They just become less visible, because when we exit telophase, we're going to go right back into interphase, because again, it is a cell cycle. So telophase's job is really to rebuild the nucleus, and the nuclei really in this case, because we have two of them. So by the time we finish telophase, we should see two distinct nuclei completely formed. Nuclear envelope and everything. Now, the other part of all of this is that we do have cytokinesis. So this part has been the division of the nucleus, which is the mitosis or karyokinesis part. Cytokinesis, if we take just a moment to mention what cytokinesis is going to do, for cytokinesis, it starts in this phase. So cytokinesis usually starts here. And the reason we know that it is starting is because we had that elongated cell. So this was indicating that the cell was kind of stretching itself out. And really we have special spindles here, the ones that are not attached to the chromosomes, that are participating in this and helping it to kind of stretch out. And then over in this case, I didn't draw it at the moment, but if we're talking about an animal cell, and I just alter my picture here, what's going to happen here is we're going to start to see that these cell membranes, the plasma membrane, starts to pinch in. And that is just a continuation of the cytokinesis process. So this will eventually really pinch all the way together, so that we'll take that one cell, and that one cell will turn into two separate cells. These are our daughter cells. They each have their own nuclei inside of them. So typically, by the time telophase ends, cytokinesis has ended as well. So they do tend to overlap a lot in their time frame. Cytokinesis did not just split the cell, but it also split all that stuff that was in the cytoplasm, which would include a lot of different organelles that we have present there and various chemicals that would also be present in the cytoplasm.